Hello students, myself Om Prakash Katre and you are welcome to Biology Reward. Today we are discussing class 12th chapter Molecular Basis of Inheritance and our topic is the lac operon, or this is also inducible operon. So students, lac operon, this is the mechanism of gene regulation. The genes which are present on the DNA, these genes are determining the characteristic of the organism. So in 1961, the regulation of the protein synthesis in E. coli elucidated by the Francis Jacob and Jacques Monod's. So this model lac operon is also called Jacob's and Monod's model of gene regulation, and this is given by both scientists in 1962. So regulation of protein synthesis in E. coli, it means prokaryotic, is explained by Francis Jacob and Jacques Monod in 1961, and this is called the lac operon model. So it is the regulation of gene in prokaryotic. Students, lac operon word is used. Lac means what? Lac it referred to lactose. It is the sugar. So lactose, lac is referred to the lactose. And another word is operon. What is the operon? It is segment of the DNA. So operon is a segment of DNA. It has polycistronic structure. It means there are many genes. So one segment of the DNA where are many genes. So prokaryotic, as we have discussed, prokaryotic, they have the polycistronic gene. So the lac, what is operon? Operon is a segment of DNA. It has polycistronic structure gene. So there are the many genes regulated by common promoter and regulatory genes. Okay. So lac operon, lac is referred to lactose, and operon is a segment of DNA. It has the many polycistronic genes which regulated by common promoter and regulatory gene, and this one expression is in the prokaryotic. Second, generally the operon is generally off. It means their synthesis of mRNA it's not take place in generally. The operon is generally off. There is not transcription. Transcription, it means DNA is changed into the RNA or transcript into the RNA. So there is not transcription, no formation of enzyme or protein. So we should remember lac operon model given by Jacob and Monod in 1961, and this is the regulation in prokaryotic. Lac refer to the lactose, and operon is a segment of the DNA has polycistronic gene structure which are regulated by common promoter and regulatory gene. Then operon is generally off. Operon is generally off, and there is no transcription. It means mRNA synthesis. mRNA is not synthesized, and no formation of enzyme or protein. So in operon, generally it's remain off, and synthesis of mRNA or synthesis of protein is not take place. We are talking the lac operon, it means lactose operon. Lactose is an inducer. It means when lactose is mixed or lactose is introduced in the system, then what happens? If the lactose is induced, it induces the mRNA synthesis and protein synthesis. So it is called, lactose is called the inducer. So when lactose is added or lactose is present, then what happens? Lactose is act as an inducer. It added in the E. coli that induces the synthesis of some enzyme. They are called inducible enzyme. So in system, when the lactose is added, then what happens? E. coli synthesizes some enzyme, they are called inducible enzymes, such as B-galactosidase enzyme, permease enzyme, trans-acetylase enzyme. They close to each other and link. It means this gene, these enzymes are synthesized in operon when lactose is present. In the presence of lactose acts as the inducer, and in the presence of lactose, E. coli, it synthesizes the inducible enzyme B-galactosidase, permease and trans-acetylase. One more important thing is there in lac operon, RNA polymerase enzyme. RNA polymerase enzyme help in transcribing the operon. It means the DNA segment, it transcribed to RNA, and for formation of RNA, which enzyme is involved? RNA polymerase. So RNA polymerase help in transcribing the principle. So 5 points are very important. Jacob and Monod gave this model in 1961. Lac referred to lactose. What do you mean by lac? Lac is the lactose, and operon is a segment of the DNA has many polycystinic genes which are regulated by common promoter and regulatory gene. Generally, the operon is switch off. It means no transcription take place and no protein synthesis take place. And third one, lactose acts as the inducer. Lactose is an inducer. If lactose is added in E. coli, it synthesizes the inducible enzyme. It is called B-galactosidase, permease and trans-acetylase. And next point, RNA polymerase help in transcribing the operon. So these are the some important finds regarding the lac operon. We have to discuss now structure of lac operon. So structure of lac operon, as I have discussed, the Jacob and Monod, they have given the structure of lac operon. So, and their mechanism of gene regulation in prokaryote. Now we have to understand what is the structure of operon. As I told you, what is operon? Operon is a segment of DNA. In segment of DNA, here you can see structure of lac operon. So in structure of lac operon, these are the important structure. Regulatory gene, which is indicated by I. Promoter gene, which is indicated by P. P is for the promoter. Operator gene is indicated by O. Structure gene are Z, Y and A. So let's you understand first. This is the small segment of the DNA. And this segment of the DNA, it's called the operon. This operon, there is a regulatory gene. Regulatory gene is represented by I. So I is a regulatory gene. So one gene is there. Operon is polycystinic in prokaryote. So in operon, it is the segment of DNA. In this case, I is what? A regulatory gene. It is represented I. It's a regulatory gene. What happened? It code for repressor of lac operon. It means this regulatory gene I synthesizes the one protein, and this protein we used to say repressor. This is called the repressor. So repressor protein is synthesized by the gene, regulatory gene. We should remember regulatory gene represented by I, and it synthesizes the protein. It is repressor protein. This repressor protein is synthesized. It means regulatory gene, it code for repressor protein of the lac operon. Second, the regulation of lac operon by repressor is referred to negative regulation. Look at here. If regulatory gene synthesizes the protein which is called repressor protein, and due to the repressor protein, the transcription not occur. Protein synthesis not occur. And that's why the regulation of the lac operon by repressor is called the negative regulation, because due to the repressor, active repressor, protein synthesis not take place or mRNA synthesis not occur. So regulatory gene is produce the protein, it is called repressor protein, and due to the repressor protein, if it is active, in such cases mRNA synthesis and RNA or protein synthesis will not take place, and that is called negative regulation. So the regulation of lac operon by the repressor refer to the negative regulation. Why? Because active repressor do not allow the RNA polymerase to synthesize the mRNA and protein, and that's why it is called negative regulation. So this is I. Second, in operon, second gene is promoter gene. So this promoter gene is indicated by the P. Promoter is a P. Promoter gene, what is the function? RNA polymerase bind and transcription of mRNA start. So during the mRNA synthesis, this promoter side here, RNA polymerase enzyme, this is the enzyme's name is RNA polymerase. So this RNA polymerase enzyme is bound with the promoter, and if RNA polymerase bind with the promoter, transcription of mRNA start. So promoter gene, P gene or promoter gene, here this gene is provide site for the binding of RNA polymerase, and then protein synthesis or process of mRNA is start. So this is P gene. Another is operator gene. Operator gene is represented by O. So operator gene function, it controls the functioning of structure gene. We'll see that here some genes are located, they are structure gene, and the structure genes are synthesizing the some specific enzyme, and that enzyme help for the process of mRNA synthesis. So this operator is a main controller of the operon. So I is regulatory gene. Okay. Operator, it controls the functioning of structure gene, one function. Another there are structure gene, and this structure gene we can say lac Z or gene Z only. There lac Y, gene Y, and another lac A. It means this is gene A. So these three lac Z, Z, Y, A, these are called the structure gene. So what does now structure gene, what is the function? First understand. There are three structure genes in operon. Structure gene, they synthesizes the enzyme and help in synthesis of mRNA. So we have to discuss I is regulatory gene, and it produces the repressor protein. And due to the active repressor protein, synthesis of mRNA will not take place because RNA polymerase will not join here. Second, P, its promoter, and its here RNA polymerase is bound and transcription of mRNA takes place. O is for operator gene, and it is the control, the functioning of the structure gene. This structure gene, Z, Y, and A, this functioning of the gene is controlled by O. O is for operator. Now, structure genes, there are three structure genes. One of the structure gene is Z. So, Z is for, it code for B-galactosidase. The Z gene is called B-galactosidase. And due to the B-galactosidase, it synthesizes, Z gene synthesizes the B-galactosidase. And due to the B-galactosidase, it lactose change to galactose and glucose. This is the very important. What is the function of Z gene? It synthesizes the B-galactosidase and it converts lactose into galactose and glucose. So this Z gene synthesizes B-galactosidase, and this is help for changing lactose into galactose and glucose. So this disaccharide sugar change into the monosaccharide, so galactose and glucose. Second, this is why it synthesizes the permease enzyme. It codes for the permease enzyme. Code it means synthesize permease enzyme. What happens? Increases the permeability of the B-galactosides in the cell. Increase the permeability of cell to B-galactoside. So B-galactosidase permeability is increased in the cell due to the permease enzyme. And A is synthesizes the enzyme. It is thiogalactoside transacetylase. Thiogalactoside transacetylase. We should remember transacetylase. So this transacetylase enzyme, what happened? Removal of toxic thiogalactoside. So we have three structure gene. Z synthesizes the trans B-galactosides, which convert lactose into glucose and galactose. Y synthesizes permease enzyme, and it increases the permeability cell to B-galactosidase. B-galactosidase is easily introduced in the cell. And transacetylase, this A gene transacetylase, it removes the toxic thioglycolate. So this unit where I, P, O, I is regulatory gene, promoter gene and operator gene, and Z, Y, A, these are the structure gene. So these all three are controller genes. So it is regulatory gene. Here you can see this unit also. So regulatory gene is having the promoter site. So in our book P is also written. So it is the promoter site of regulatory gene. So promoter site of the regulatory gene. Now I stand for regulatory gene, and regulatory gene synthesize repressor protein and active repression protein. This mRNA synthesis is blocked or mRNA synthesis not take place, and so it is called negative regulation. P is stand for promoter gene, and this promoter site RNA polymerase bind and transcription of mRNA start. This is the structure of promoter. Operator G, O, and this operator it controls the functioning of this all structure gene. There are three types of structure gene which synthesizes the enzyme. Z gene code for B-galactosidase, and this B-galactosidase enzyme convert lactose into galactose and glucose. Y, it synthesizes permease enzyme, and it increases the permeability of the cell to B-galactosidase. And A is synthesized the transacetylase enzyme. It removes the toxic thioglycoside. So this I for regulatory gene, P for promoter, O for operator gene, and ZYA, these are the structure gene. So this is complete operon unit. One marks question is asked to you, what does operon consist of? So operon consists regulatory gene, promoter gene, operator gene and structure gene. Now we have to study here, as I told you, lactose is an inducer. Lactose is an inducer. It means it induces the process of protein synthesis. So what happens in operon in absence of lactose? So in absence of lactose, I, it is P, promoter, operator, and Z, Y, and A. These are the structure gene. In absence of lactose, if lactose is not there, so here absence of lactose. Without lactose, what happens? This regulatory gene synthesize the repressor. So regulatory gene synthesize one enzyme and it form the protein, and this we used to say repressor protein. So repressor repressor is produced by the regulatory gene. This is the repressor. So in absence of lactose, regulatory gene, it produces the repressor. Regulatory gene, it produces the repressor. Now this repressor bind to operator. This repressor, it bind to operator. Here it bind. Repressor is bind to operator. So when the regulatory gene synthesis the protein, that is repressor, and this repressor bind to the operator site. In such cases, turn off. It means RNA polymerase is remain off. RNA polymerase will not start the protein synthesis. In such cases, RNA polymerase stop transcription. So, when repressor bind with operator site, RNA polymerase stop transcription. And when RNA polymerase stop transcription, it means mRNA synthesis not take place. The structure gene ZYA do not produce mRNA, enzymes and protein. And in such cases, simply in absence of lactose, repressor produced by regulatory gene bind with the operator site, RNA polymerase will not work, and here no transcription. No transcription not take place, or we are saying no synthesis of mRNA. It means no synthesis of protein. Protein synthesis will not take place because mRNA is not transcribed. So it occurs in the absence of lactose. Again, see, in absence of lactose, regulatory gene produce the repressor protein, and this repressor protein bind with operator, and even it bind with the operator, RNA polymerase will not work, and in such cases no transcription. It means no synthesis of mRNA and no protein synthesis occur. So it is the regulation of the gene by the regulator, and that is called, here mRNA synthesis is not take place, so the regulation of operon by the repressor is said to be negative regulation. Second part is after the in presence of lactose. Suppose the lactose is present. In case of where the lactose is present, so it is operon unit. In this case, lactose present. Lactose presence. In presence of lactose, what happens? This I, it means regulatory gene will synthesize the repressor. Repressor will always produced by regulatory gene. So here we have studied in lactose is an inducer, and here you will see it's a process of gene regulation or mRNA synthesis generally or normally is remain off. So why is remain off? Because the regulatory gene is always synthesizes it's a repressor. So this repressor. But now lactose is present. So here it is, I am mentioning it is the lactose. This is the molecule of lactose. So if lactose is present, in such cases what happens? This repressor protein get inactivated by the lactose. The lactose combining with the repressor. Here regulatory gene synthesize repressor, and when repressor is produced, here you are adding the lactose. It means in the presence of lactose. Now what happens? Lactose combined with the repressor. Our repressor is inactivated here. Due to the lactose, the repressor get inactivated. And if repressor is get inactivated, now it will not bind with the operator. So here lactose bind to repressor. Lactose is bind to repressor. Repressor is inactivated. This repressor now become inactive. In such cases, RNA polymerase will combine here. Is the RNA polymerase. It is the site where RNA polymerase will combine. So it is RNA polymerase. And when RNA polymerase Now in such cases, RNA polymerase combined with the promoter site. What happened? Operator turn off. It means this operator site is off. Operation site is on, and its operator give command to the promoter or synthesis of this mRNA. So when lactose bind with the repressor get inactivated, operator turn on is become on, RNA polymerase start transcription. So RNA polymerase will combine with the promoter site, and here transcription is started. Structure gene produce enzyme. So these enzymes, structure gene, that Z gene synthesize big galactosides. It synthesizes the big galactosides. Y permease and A transacetylase. So when repression not combined with the operator, because this get inactivated by the lactose, and in such cases operator gene become on, and due to this one RNA polymerase start transcription, and structure gene Z synthesize big galactosides, Y synthesizes permease, and A is synthesized transacetylase. So formations synthesis of enzyme take place, and this enzyme help for the synthesis of mRNA, and later it help for the protein synthesis. So this is called turn on. So if lactose is present, the lac operon is on. If the lactose is absent, the lac operon will be off. So total we will see here two important things in operons are: number one, each operon is a segment of DNA is having polycistronic, and there are I regulatory gene, P is for promoter gene, O operator gene, Z is a structure gene, Y is also structure gene, and A is also structure gene. So this structure gene, Z synthesize big galactosides, Y permease, and A transacetylase. So what does the operon consist of? So regulatory gene, promoter, operator, and structure gene. Then regulatory gene synthesize the repression protein which combine with the operator site, and process of transcription is blocked. So it is called negative regulation. Promoter gene, it provides site for RNA polymerase, and it starts the transcription. Operator gene, it controls the functioning of the structure gene. And structure gene, as I told you, Z synthesize big galactosides which convert the lactose into galactose and glucose. Y code for permease and increase the permeability of cell to big galactosides. And A is synthesized transacetylase which remove the toxic thioglycan. In absent of the lactose, I synthesize the repressor protein, and which combine with the operator site. So polymerase, RNA polymerase stop working, no transcription occur, mRNA synthesis and protein synthesis is not occur when the absent of lactose. But when lactose is present, in such cases, lactose is combined with the repressor protein, and repressor protein get inactivated. In such cases, it will not combine with the operator site. So operator switch on, and it give command to the promoter, and RNA polymerase is combined with the promoter site and start the transcription. And that time structure genes, Z synthesize big galactosides which convert lactose into galactose and glucose, and Y synthesize permease which increases the permeability of the cell to galactose, and thus the structures gene, they form the enzyme, and that enzyme help for the mRNA synthesis and protein synthesis. So it is all about the lac operon model of regulation of gene in prokaryotic.